Yes, I will also be talking about microbial ecology, or specifically the ecophysiology of multicellular cable bacteria. So my focus will be solely on cable bacteria and all the insights, or some of the insights we got from stable isotope probing combined with nano sims, which was a large part of my PhD research. I will start with a short introduction of what cable bacteria actually are, because they're very famous in a small community, but not in a large community. And then I will show some of the work we've done with stable isotope probing and the nanosims. And I will specifically focus on the division of labor and growth within a multicellular cable bacteria. And I will show some highlights of the polyphosphate dynamics that we discovered. So here you already see some of the nanosims imaging. So you can see that the cable bacteria are multicellular. You can see multiple cells. So here you see 5 filament segments, so 5 different filaments, and then segments of it. And here you see one of the images from the polyphosphate story. So again, you see many different filaments in one region of interests. So what are cable bacteria? They live usually in marine sediments, although they have also been found in freshwater and aquifer sediments. And they are centimeter-long filamentous sulfide oxidizing bacteria. You can see a fish image here, so you can see clearly that they are multicellular. Here you actually see a really nice image of a sediment core. This is the Oxic iron-rich sediment that has been lifted up. And here you see the black sulfide-rich sediments, and in between you see all these fiber structures that are cable bacteria. So this is an image that is taken with a regular camera, so you can actually see them in real life if you look real close. And they have a really special capacity because they can conduct electrical currents over longer distances, so millimeter to centimeter scale. And they use conducting fibers in the shared periplasm between all the cells, which you can see here, to transport these electrons. So they have a very peculiar energy generating metabolism. Here you see an image of a cable bacteria in the sediment. The sediment has an oxic zone. Then below the oxic zone, you have a so-called suboxic zone, and then below that you have a sulfidic zone. And the cells in the suboxic zone are oxidizing sulfide via this redox half reaction. So this half reaction, this generates electrons, which you can see here. These electrons are transported upwards towards the oxic zone, where the cells in the oxic zone use these electrons to reduce oxygen. So what we have in one organism, we have spatially segregated redox half reactions. So all these cells within the filament are cooperating for their energy supply. So there is a clear division of labor between the cells residing in the oxic zone and the cells in the suboxic zone for energy generation. And for this presentation, it's important to know that usually around 5 to 10% of the cells are residing in the oxic zone. And these filaments can move in and out of the oxic zone. So they have some sort of gliding motility. So we were really curious how the electron flow, so how the energy or the electron flow is coupled to energy conservation and biosynthesis. And we did this by using stable isotopes. So we had an enrichment culture in the lab for cable bacteria. This is just sieved sediment where cable bacteria once were found. And we subjected the sediment to all their favorite favorite environment, and then you have to wait a few weeks, and then you have a cable bacteria culture in the sediment. We injected the sediment with labeled inorganic carbon, so 13C bicarbonate in our case, and 15N labeled ammonia. And then after 24 hours, so after a labeling period of 24 hours, we picked filaments from the oxic zone and from the suboxic zone. So you can handpick them with a glass, with a glass hook. And then we visualized them with scanning electron microscopy with SEM. And then they were measured with nanosims to see if they had any label, if there was any label uptake during this 24-hour period. We also measured the labeling grade of the pore water. So we measured the background labeling of the pore water with isotope ratio mass spectrometry. So we could do some calculations on the carbon assimilation and the ammonia assimilation. So after we have prepared all the samples and measured them, we got these types of images. So here you see 4 nanosims images from where you see the 13C atom fraction. So it's 13C over total C. The first image is from filaments that were retrieved from the Oxic zone. And the right 3 images were from sediments retrieved from the suboxic zone. And as you can already see, the first image, in the first image here, that they didn't take up very much carbon, their 13C bicarbonate, which means that they were not assimilating any carbon in the oxic zone, whereas they were assimilating carbon in the suboxic zone. If you zoom in on each of the single filaments, you see that among the cells in the filament, the uptake is pretty homogeneous, whereas there's a large difference in uptake between different filaments. So nanosims is a really good method to investigate phenotypic heterogeneity, because they were all subjected to the same environment, but the outcome between filaments is very different. And we also did a labeling with 15N ammonia, so we measured this as well. And these are the results from the 15N ammonia, atom fraction, or so 12C 15N over total CN. And if you compare these images, they appear to correlate very well. The scales are slightly different, but high bicarbonate uptake means high ammonia uptake. So we did these measurements for many different filament fragments, which you can see here. So here you see a graph. On the x-axis, you see the 13C atom fraction. On the y-axis, you see the 15N atom fraction. The dotted line here and here are the natural 13C and 15N isotope ratios. And here these lines represent the labeling, the 13C atom fraction of the pore water, so the background label. Here you see all the squares were taken from filament fragments from the oxic zone, all the circles from filament fragments from the suboxic zone. So each data point represents the mean 13C and 15N atom fraction for a filament segment, which is usually between 5 to 10 cells, although from some filaments we took multiple measurements along the filament, so this could be some more cells. Well, what we could conclude from this data already was that there was a strong correlation between carbon and ammonia uptake. And if you see here, all the squares grouped together at around the natural ratio. So there was little or no 13C and 15N enrichment in filaments or cells from the oxic zone. Because we had we have the 13C atom fraction of the pore water, we could do quantification with these nanosims data. So we could convert the 13C atom fraction to inorganic carbon assimilation rates, which is what you see here. So here you see the assimilation rates from different filaments in the oxic zone, and here from different filaments in the suboxic zone. And this made it very clear that biosynthesis and growth only happens in the suboxic zone. Which is where we see carbon and ammonia uptake, and the carbon assimilation rate is highest in the suboxic zone. Strangely enough, we did not see any biosynthesis and growth by cells from the oxic zone, so cells that were performing oxygen reduction. Which led to the conclusion that cells that are doing the oxygen reducing half reaction cannot generate energy. which is strange because you need energy to survive, but it is in accordance with genomic and proteomic data on cable bacteria genomes and proteomes, where they found no known terminal oxidases, which are proteins that can couple oxygen reduction to energy generation. So nanosims is completely complementary to these omics methods, where the omics can actually be used to generate hypotheses that can be tested using stable isotope probing in combination with nanosims. So from this, we have gained insights into the division of labor within a filament. So here you see a filament. The green cells are in the suboxic zone, the red ones in the oxic zone. And cells performing the oxygen consumption cannot generate energy, so they cannot grow or divide. So they perform some sort of community service for the rest of the filament. Because if a filament doesn't have access to oxygen, it cannot get rid of the electrons, so it can also not oxidize sulfide. So this gave us the next question: how do these cells survive in the oxic zone? Because you need energy to survive. And we have a hypothesis, or we had an hypothesis, that they rely on storage compounds such as polyphosphate granules. And polyphosphate granules are found everywhere in cable bacteria. So here you see a DAPI image, and all the bright spots are polyphosphate inclusions. Here you see a SEM image, and all the bright white spots are polyphosphate inclusions. So polyphosphate is a biopolymer. It's a polymer. It is at least 3 phosphate groups long, but it can be up to 1,000 phosphate groups long. And it's a very old molecule, and it can have several functions in a cell. So as you can see here, the bond in polyphosphate is the same as the phosphoanhydride bond in ATP. So it can be a substitute for ATP, although the energy release is slower. But it can also be used as an adjustment for survival in the stationary phase, or to protect cells against oxidative stress, so stress in oxic environments. So we found them in almost all cable bacteria cells, and it doesn't matter from which redox zones we retrieve these filaments. So they're found in the oxic zone, and they're found in the suboxic zone. So we wanted to know, what is the role of these polyphosphates? So we performed another labeling experiment, followed by nanosims. This time we labeled with 18O labeled water, and again with 13C labeled bicarbonate, to see which filaments were metabolically active. After 6 and 24-hour labeling, we cut the course. So we divided them into the oxic zone, the transition zone, which is the zone a millimeter below the oxic zone, and a suboxic zone. So this transition zone is actually the top of the suboxic zone. And then we handpicked filaments again and measured them with SEM, but mostly with nanosims. So we got now 18O atom fractions and 13C atom fractions. And 13C shows general metabolic activity and growth, whereas the 18O labeled water exchanges oxygen atoms with phosphate in the pore water. So you get a pool of 18O labeled phosphate, which can then be added onto the polyphosphate chain. So if you see 18O labeled polyphosphates in your cell, you know that polyphosphate chain was synthesized during the incubation. So 18O labeling in the polyphosphates shows synthesis of that polyphosphate. And although there are many microscopic methods to look at polyphosphates, so you can use, for instance, you can use FISH or DAPI or other fluorescence microscopy, or even scanning electron microscopy to look at polyphosphates. This is the only known method to assess polyphosphate activity, or at least synthesis. Well, we wanted to know, what is the effect on the redox environment, and is there a variation in the 18O labeling of polyphosphate within a cell? Sometimes there's multiple polyphosphate inclusions in one cell within a filament. and between or among filaments. So we got some really nice data. Here you see the secondary electron image from the nanosims. So you see 5 filament segments. These are from the suboxic zone. And here you see the corresponding 13C atom fraction image. As you can see, 2 of these filaments did not grow during the incubation period, whereas 3 filaments did show growth during this incubation period. And here you see the corresponding 18O atom fraction and the relative phosphorus content of these cells. And as you can see, there's a clear correlation between the phosphorus content and the 18O labeling, where the 18O labeling shows that these polyphosphate inclusions were partly synthesized during the incubation. So in previous experiments, we found that in the suboxic zone, about half of the filaments was not actively growing. And in this experiment, we see that still a large fraction is not actively growing, but most of these cells did assimilate or did synthesize polyphosphate. So they must have had access to energy to synthesize these polyphosphates. So they were doing something. They're not dead or not doing anything. For this particular region of interest, we measured a lot of planes, more than 7,000 planes, which we could stack. So we sputtered all the material away, but this allowed us to do some 3D imaging or some depth analysis. And here we show 2 of these depth analyses. So here you see this inactive filament, or at least that didn't grow. And we do a depth analysis, a lateral depth analysis here. And you see here the 13C atom fraction with depth, the 18O atom fraction with depth, and the relative phosphorus content with depth. And you can clearly see these polyphosphate inclusions in the cell. And you can see that they take up a large part of the cell volume. The same actually holds true for one of the active filaments. So this is this filament. It's the lateral analysis along this depth, or along this filament. You see the 13C atom fraction with depth. You can actually see some the beginning of cell division here in this filament. And here you can still see the in the 18O labeling, you can still see the polyphosphate granules as a more bright spot. So this means they build up a lot of polyphosphate during the incubation. And then here we see some representative images from the nanosims from each of the zones. So the oxic zone, the transition zone, and the suboxic zone after 6 hours of labeling and after 24 hours of labeling. And we could conclude that between filaments there's a large variation, whereas within a filament, so among cells within a filament, there's a small variation, also in the polyphosphate buildup. And we could group all the filaments in 4 groups, where the first group only contained a very few filaments, so that didn't take up any, didn't grow, so didn't take up any 13C label and no 18O label in the polyphosphates. A few filaments showed only 13C labeling, so no polyphosphate labeling. And then there's a large fraction, about a quarter, that didn't grow, but these filaments did show polyphosphate synthesis. And about 70% of the filaments showed both growth and 18O labeling in the polyphosphate. So both growth, both polyphosphate labeling. These are all from filaments from the suboxic zone, and the transition zone. So what we see is that 92% of the filaments showed polyphosphate synthesis, even though some of them didn't show any growth. And what we could see, even in the oxic zone, we could see 18O labeling of the polyphosphate granules here, in both after the 6-hour incubation and after a 24-hour incubation. which means that these cells from the Oxic zone must have had some access to energy during the incubation. We know that cable bacteria move around in the sediment, and working from the hypothesis that they cannot generate energy in the Oxic zone, we hypothesized that these cells migrated from the suboxic zone to the Oxic zone. But since we do not see any carbon labeling in these filaments, this means that before they migrate to the Oxic zone, they stop growing, or they shut down growth, start synthesizing polyphosphate, and then move up into the Oxic zone. So they are sort of prepared to go into the Oxic zone, and they need the polyphosphate to survive in the Oxic zone, either because they can use it for energy, or they can use it for protection against oxidative stress. Also in the suboxic zone, we see many filaments that didn't show growth, but did show polyphosphate synthesis. And this is the first insight into what cable bacteria are doing during periods of non-growth, which is a mystery for many bacteria. So this labeling gave some insights into the periods of non-growth. So by using stable isotope probing and nanosims, we have both qualitative and quantitative insights in the metabolism of the cable bacteria. And we also got insights into the function of subcellular structures, in our example, polyphosphate granules and element fluxes within a cell. So this couldn't have been done by any other method. And obviously I didn't do this by myself. So these are all my collaborators, which need to be acknowledged. And that's it. If you have any questions, let me know.